I'd like to invite Slava Tkachenko up to talk to us about how public good producing cells could invade a tumor. Another awesome Cleveland scientist, just throwing it out there. We have a huge, wonderful network of evolutionary scientists brewing in Cleveland, right? It's amazing. Cleveland rocks.
Okay, while slides are being pulled up, I must admit I thought I would talk later, but David was correct. I'm talking right after him. By the way, David, no thanks. I thought you promised a talk as bad as you could get it. Was that your best shot at bad, or you just lied to me? Okay. Okay. Speaking of which, so green forward, red backwards, and no pointer, I guess. Okay, so this would be the first talk on one specific project, which I did in McFarland Lab. By the way, sorry, I'm Svyatoslav Tkachenko, should have introduced myself. And in collaboration with great Michael Hinchevsky from physics department, we are studying how whether public good producing clones can invade a tumor population. So in this context, I want to say public goods are any diffusible factors that can benefit cells around. And this is not quite straightforward intuitively, because someone tries to invade and then someone is producing something good for everybody. You know, this old ecological altruist problem. Will it be able to invade? I've read some hand-waving argument that not, but we'll see. So a little outline. First, I'll discuss intertumor heterogeneity and clone coexistence. This will be an introduction, which everybody here probably knows better than I do, but I'll present. And then about our approach. First about the numerical simulation, then about the analytical approach. I like outlines so much that I gave the introduction a separate outline. ITH and its implications, how clone coexistence points were found in replicator dynamics, and in general possibility of clone invasion. Okay, everybody in the audience probably knows more about this than I do, so I will understand if you yawn, but I will appreciate if you don't, because I needed some cohesiveness in this talk, so I'm doing the introduction. First of all, it's an accepted fact, and you even heard about this intertumor heterogeneity today already. It unfortunately correlates with poor prognosis. It is implicated in treatment resistance, and we did here and yesterday at the poster session, you saw about those little clones hiding somewhere in plain sight that carry resistance, which might explode once the other ones that are more fit are pounded down. And ideas of using ITH against the tumor itself. Once again, we heard about this today. We've heard about adaptive therapy at the poster session yesterday. So in a nutshell, just apply therapy, pound on the more fit clones. Then when drug-resistant clones start taking over, remove it. So the fitter under normal conditions will try to take care of them. So some approach, okay, at least one of the approaches of how clone coexistence points were found. Jake already talked about stable or unstable equilibrium. It's mostly about stable now. I'll talk about replicator dynamics approach, and about some problems with it. First, replicator dynamics. In a nutshell, it's very simple. One literally little with 4 arguments in it, differential equation. So how fast population of type I changes depends on the population itself, its size at the moment, on how much benefit it gets, its fitness, and the fitness is relative. So those brackets could be actually one term, something like delta pi. Everything is relative here. If you're pretty crappy in one surrounding, you can be the boss in another surrounding. But devil's in details as usual, because finding that fitness is not straightforward, and it might be changing dynamically. But people use this approach and get some nice results. After all, with these differential equations, we get nice averages. And just to give you an example, some paper from 10 years ago, and there are papers later, but here the picture was the most intuitive I could find. Arcadi assumed a certain shape of the benefit function. It was sigmoid, surprise, surprise. And he calculated some parameters dependent on it. The upper is cost of production and how it depends. You see different curves, no pointer, out there for different costs of production. Then dependent on the diffusion range, and then dependent on the parameters of his benefit function, inflection and steepness. And you can see here how fraction of producers start from some initial population changes across generations. One more thing that I want you to remember for the next slide would be that initial fraction of producers is quite sizable, relatively speaking. It's not zero. It's not at number of producer cells being 1, as you have when an invasion starts happening. It'll be useful in a second. So there are some problems with this differential equation approach. It's usually used for very large, well-mixed populations. But spatial structure has been shown to be important. Although well-mixed are somewhat easier to treat and people like to start there. And if you need to operate in a regime where stochasticity is important, differential equation averages might not be very useful. And that's what I was talking about in the last slide, that we're not at n equals 1 there. Because random statistical fluctuation, not described by some average in differential equation, oops, one cell is gone, it's nothing. But if one cell is the whole clone, it's something. It's gone. So we try to circumvent these problems in our approach. First, the numerical simulation. We started with one mutant cell shown to the very left as a red dot on a lattice with von Neumann neighborhood, square lattice with von Neumann neighborhood. Then it produced some public goods, which diffused away. Did diffusion by random walk with absorbing boundary conditions. Once again, we try to respect stochasticity as much as possible at every step. Hence, no simplifications. Then we did some binding, once again, respecting stochasticity using the Gillespie approach. And as you can see by smiley faces on the third panel, some of them did bind some goodies. Great. And at the end of the day, literally at the end of the day, the proliferation happened using the right Fisher approach. Basically, every cell produced offspring in numbers proportional to the fitness it obtained. And number of bound goodies were mapped to the fitness using a Hill function here. Is that all I want to say here? Okay, I'll run out of time so soon, so let's stop here. Okay, this did give some results. Please, I don't know why 2 of the panels are white here. They used to have some color to them on my computer. Okay, whatever, just ignore them, please. Sorry about that. And please ignore gray cells on those heat maps, because with this run for purity, we changed the code and I ran it again, and those gray cells are for the parts of the parameter space which didn't finish by the time I was putting together this picture. So on the left half would be different values of K off in different panels. K on versus D, where D is diffusivity. K on is the association constant, K off is the dissociation constant. And we went for the biologically appropriate values of all the three parameters. And on the right, where you cannot see 1/3 of it, it's each panel is a different K on used, and it's K off on the horizontal axis, diffusivity on the vertical axis. But anyway, the more important, not more important, but what is easier to see is on the left anyway. You see the trend that when you go to larger K-ons and lower Ds, you get a larger invasion probability. Unfortunately, on the lower panels is the slowest part of the simulation, so we have more gray there. But it's redder. So when it's red, it's invaded. And K off versus D, K off also has some effect, but it's smaller than K on. And sorry, you cannot see some of them, whatever is happening. And speaking of D and K on working together, we could put them in the same variable, which is D over K on, and we got a nice sigmoid across different parts of the parameter space. And it makes sense that they work together because D and K on are kind of doing the same thing in this approach. D is diffusivity. When you're flowing slower, you give a cell around which you're flowing a better chance to grab you. And K on acts in the opposite direction. When K on is good, the association constant, the cell is more prone, it's more hungry. It'll grab you faster. Okay, now the analytical approach. The approach was somewhat different. Here we're using radial coordinates. So producers in the middle, non-producers around. And reaction diffusion equation, this is basically a regular diffusion equation with a sink and a source. Here, diffusion of free molecules. This is the source, production of PGs, with rho being their density and delta how they produce, minus absorption. This is the sink. And U of R is the concentration profile. We can solve the equation for it, and we'll use this normalized form, PR. And in Moran approach, which was used by Mike, fitness is there, okay, fitness depends on bound goodies, which depend on free goodies, which is PR over there. And S helps map bound goodies to benefits. Moran gamma in this case is, okay, superscripts went to subscripts, but they're still there, good. Fitness of non-producers over the fitness of producers. And probability of one producer to go into M producers is the usual Moran formula. There's basically a 1 in the numerator. And in the analytical approach, so you can rewrite it, look for limiting cases. For delta going to 0, where delta is delta D over K on, you get the usual Moran fraction, which is on top. And when delta goes to infinity, you get a neutral case. And lo and behold, although approaches are different, right, Fisher versus Moran, square lattice versus radial, they do map to each other pretty well. The horizontal axis was rescaled to account for a bunch of stuff, but in general, they're on top of each other, which is not too bad. In conclusions, I'm out of time, so I think I'll stop here. You can look at the conclusions, but I gave you the main one. They do seem to be able to invade, and analytical and numerical approaches map to each other well. Sorry, I did take more time than I should. Okay, McFarland lab.
So in many cases of in tumor, this common good involves not a direct common good, but mediation through microenvironment, for example, angiogenesis. So it's a question like if a cell upregulates expression of, say, angiogenic factors, how can it invade? So have you looked into looking at this public good invasion in the context where
it's mediated by a third party, when the common good is mediated by the third party, like angiogenesis, and there are other examples.
Yeah, I understand what you're talking about. Funny enough, when I started this, I was using specific K on, K off, and D for VEGF, which doesn't act directly, but it's doing exactly what you said. I didn't do that. It might be in future direction to investigate how close that would relate, whether we need to introduce additional steps. But my first thought about this is that the same machinery can be used for describing it. Just this K on, K off, and D might be taken to different parameter regimes, because after all, you can, like diffusion will be on the order of days maybe, because everybody gets benefit, not because some diffusible factor flows somewhere, but because the capillary is created there, and then everybody benefits. So it'll be a very different parameter regime, but I think this can be taken to exactly the same, but with some abstract parameters, not what I was trying to use as biologically relevant, basically from measurements. I took ranges of K on, K off, and D. But yeah, sorry.
Any other questions? I think we're actually a little bit behind, so maybe we can thank Slava again.